All right, let's go ahead and get started with introductions. So we've got a lot to cover today, so I'll keep letting folks in from the waiting room as we go through this, but welcome. I'm Caroline Sell from Future Harvest. I'm the Central Chesapeake Field Program Manager, and that means I coordinate events like this. We've got a bunch of great speakers for y'all today, but before we get started, we have a couple housekeeping things really quickly. If you're not already muted, I'm going to ask you to mute if you are not a speaker. We are recording this meeting, and so we will be uploading it to our website later, and so that will be available for you to view again. And if you look down on the bottom of the screen, you're going to see a chat box. It's a little like bubble with an ellipsis in it. I'm typing hello in the chat right now. You can use that to ask questions during the event, so please do, and we'll be answering them as they come in. This is going to be as interactive as possible. Feel free to go ahead and introduce yourself now in that chat box if you'd like as well. And we're going to have a break in the middle, so there will be a break if you need to run to the restroom or grab a snack or anything like that. And we're going to have a poll at the beginning, which I just launched, so you should see that pop up on your screen in just a second. We're going to gather a little bit of information about y'all so we have a better sense of folks' experience levels and what kinds of things you might want to learn. And then please don't leave the chat at the end. We'll be launching a post-event survey, and that helps us collect information about, how it went for you and what kind of information that you learned. All right, so without further ado, I'm gonna stop chatting and we're gonna go ahead and get started. All right. Can y'all see my screen okay here? All right, fantastic. Oops, I have bumped back from the beginning. There we go. All right, so welcome to the Hands-on Soil Test Clinic. We have some wonderful speakers for y'all today, and they're gonna do a much better job than I will of introducing themselves, so I'm gonna keep it very brief. But we've got Lisa Garfield, Amanda Cather, and Linda Belsons-Rollis. All right.
Hi, everyone. I'm Lisa Garfield. I'm the Million Acre Challenge Manager for Future Harvest. I also have been working on coordinating the Maryland portion of the Soil Health Benchmark Study, which I'll talk about a little more in a moment, and was also a farmer for many years, and a plant and soil scientist. And I'll be leading you through the soil test portion of the discussion today.
Hi, everybody. I'm Amanda Cather, and I'm the Million Acre Challenge Project Manager, Project Director. And I'm also the farmer, owner, and operator at Plow and Stars Farm since 2014. I was the farm manager at Waltham Fields Community Farm in Massachusetts for 10 years before that, and managed a couple of urban farms prior to that. I also just love soil tests, and I'm really excited to hear Lisa talk more about them because she's amazing.
Great. And hi, everyone. I'm Linda Belsons-Rollis, and I'll be talking about the compost test analysis just for a few minutes later on in the presentation. I work for the Institute for Local Self-Reliance on a Composting for Community project. Been there for a few years, and actually before that I did the Future Harvest Beginner Farmer Training Program back in 2014. So I'm an aspiring farmer, but I do have the great pleasure of working with Lisa and Amanda on the Million Acre Challenge project as a core team member. And yeah, we represent sort of the composting piece of the regenerative ag puzzle. So it's great to meet all of you today.
Great. This is Amanda. I'm just gonna talk briefly about the Million Acre Challenge. The Million Acre Challenge is a collaborative effort to advance soil health and farm resilience across Maryland and the Chesapeake region. And we're really looking at what that means for us is really trying to get farmers to make progress on their farms. Whatever type of farm, whatever type of production system, wherever you're starting from, just taking steps towards soil health on about 1 million acres over the next decade. So we just started our project last year. We have 6 founding partners: Future Harvest, Chesapeake Bay Foundation, Fair Farms, which is a project of Waterkeepers Chesapeake, the Institute for Local Self-Reliance, Institute for Energy and Environmental Research, and the Hatcher Group design firm. And we have 5 different working groups. So we're really looking at soil health from 5 different perspectives. We see farmer education, and especially peer-to-peer education, as being really one foundational piece of improving soil health across the state. Science is another piece. So Lisa will talk more about the soil health benchmark study, but that's a big piece of what we're looking at. The business case for improving soil health. So what's the return on investment for farmers for improving their soil? That's really important because soil health can seem very non-immediate, and it can seem like it's hard to see in the moment the return on investment there. So we're looking at that. Public outreach. So really trying to impress upon the public the importance of requesting and looking for, seeking out foods grown in healthy soils, and also policy. How can we create policies that provide incentives to farmers of all sizes and all production methods to work on improving their soils? And we hope that after this workshop, you'll stay engaged with us either through the Soil Health Benchmark Study. We hope that in the coming months, you'll start to see more information about our soil health farmer study circles, which will be called hubs. They'll probably be regional and kind of based on the soil health benchmark study, but also expanding beyond that. And really thinking about getting information from you and farmers like you. What do you want to learn about soil health, and how do you want to learn it? Do you want to learn it in a classroom setting? Do you want to learn it on field days, virtual farm tours, any way you could imagine? And what practices would you most like to implement on your farm or learn about if you're not currently farming? So lots more to come there, and you can get in touch with me, the email there, and also it'll be at the end again. Lisa, do you want to talk about soil health benchmarking study?
Yes, just unmuting.
Oh,
Thank you. So yeah, the soil health benchmark study is a long-term on-farm soil health data collection initiative, kind of a citizen science initiative, in that we're really asking farmers to be involved and engaged and hands-on in looking at a really broad set of soil health indicators that are provided through the Cornell Comprehensive Assessment of Soil Health. And the study was developed by PASA and is led by PASA. It started in 2006 there and has grown now to include over 100 farms. As part of the Million Acre Challenge, we were able to expand that initiative into Maryland. And so we started last year with 12 farms and are growing it to 39 farms in 2020 at the research collaborator level. And one of the unique things about the study is that it provides comparisons with like farms, partially through the Cornell test, which kind of places each indicator on a curve based on your soil type and shows you where you stand related to other farms with similar soil types. And then beyond that, by using your field management records to collect additional information like days of living cover and tillage intensity, that put your results in the context of your soil management practices. So that's what makes the study kind of unique. And because there's been a lot of interest in it and we want to be able to include as many people as possible, we are now adding a community scientist opportunity. So people that we don't have enough funds to provide all of the soil testing for can still join as community scientists and collect their own samples. If you could advance slides, Carolyn. Thanks. So as a research collaborator, we pay for the tests, but as a community scientist, you submit your own tests, but you still can participate in any of the programming, and you'll still receive the community benchmark reports. So it's a way to be involved and still get a lot of the benefits. But unfortunately, we don't have enough money to pay for soil tests for quite as many people as we would like to. So just something to keep on your radar. It's kind of a good way for you to just be tracking your own soil health based on a really nice broad set of indicators beyond standard nutrient testing, which will be the topic of today's webinar, where we will mostly be focusing on your kind of standard soil tests that talk about nutrients. Great, so yes, today's topics will include just a really quick overview of some soil basics. We're not going to get too deep into that, but a few important things to know when looking at your soil tests, and then the really practical information about choosing a lab, how to take your soil sample, things like, you know, the macronutrients and the micronutrients, pH and buffer index, all of the things you're likely to see on a standard soil test. And then we'll go through some example reports just to show you some differing results and what that could be telling you, and how you might manage your soil based on those results.
We have a quick question, Lisa, and that's how much would the Cornell soil test typically cost for someone paying for it themselves?
So to join as a community scientist, you could choose to do either the basic test for $60 or the standard test for $110. For research collaborators, we're using the standard test, but that is very expensive, I understand. So you could, if you're doing it on your own, just get the basic test. Okay, so soils are the foundation of every farm. Your soil health is critical to crop health, and so also critical to the economic success of your farm. And in addition to offering crop nutrition, healthy soils have the potential to offer a lot of ecosystem services that are really hard to quantify or put a dollar amount on. And that includes things like proper infiltration when we have extreme weather events, filtration and replenishment of groundwater resources, improved water holding capacity during times of drought, things like pest and disease regulation, and then also things like carbon sequestration. So these are huge services that we don't always think about when we're planning for crop nutrition, but are also very critical both to crop health and to farm health and ecosystem health. So soil tests are just one way that we can, you know, they're just a tool in our belt that we can use to understand what we're seeing. And we'll go through a little bit more as we go on what they can tell us and what they don't tell us. So just to put the tests into context, soils are made up of three basic components: physical, chemical, and biological. And that middle area is the sweet spot of soil health. So according to NRCS, Soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So a large portion of what we're going to talk about today is in the chemical area of the soil. It's plant nutrients and pH and buffer capacity, base saturation, things of that nature, cation exchange capacity. But we will be dipping into, they also dip into the physical, and really all of those things are present. It's really hard to separate them out, but a lot of the standard soil tests that we look at are primarily focused on the chemical. So, next slide. So an important physical feature that is not always included in a soil test, or not all of them, is your soil texture. The texture is just the percentage of sand, silt, and clay that are in the soil. Texture can't be changed, and but it will very strongly influence the management decisions that you make and how the soil behaves. So for instance, a coarse soil, so something in the very sandy range, is going to have a very low cation exchange capacity. And so it's going to have a really hard time holding on to nutrients. And so that's something you're going to want to consider when looking at the nutrient recommendations on your tests. It's also going to have better drainage, but have difficulty holding on to water. So it's really important that you understand what your soil texture is, and it will also provide a lot of context when you're actually looking at your soil test. So in the next slide, there we go. Here's just an image of the difference in the size of the particles depending on which soil type you have. Sandy soil has very large particles, whereas clay is like very, very teeny tiny particles. And what's interesting is you might think that a sandy soil then has a larger surface area, but in fact, the opposite is true. The clay soil, because there are so many of them, have a much more surface area overall, which is the reason that it has a higher cation exchange capacity. There's more area, more sites for nutrient exchange there, but we'll get to that a little bit later. These are just some general characteristics. One really kind of fun, easy, hands-on way to get to know your soil, if you're not sure what your texture is to do a very simple jar test. You can basically just, oh yes, and I see Amanda's making a good comment. I think I have it later that the web soil survey can tell you a lot about your soil, and you should absolutely use that. A jar test is you just go out, put your soil free of extra organic material into a jar about halfway, fill it up, shake it up, and then let it sit. The sand will drop very quickly to the bottom, and then the silt will take a little bit longer to settle, and then the clay can take up to a couple of days because it's so fine that it stays in suspension. But eventually you will get kind of what you see in these pictures, which is very clearly defined layers of the particles that your soil is made up of. So that's something that you can do on your own and would be good to know before even looking at your first soil test. So the next consideration is choosing a lab. Depending on what state you're in, there may be different requirements for which lab to use. Maryland, as many of you probably know, has some fairly strict regulations for nutrient management plans and require that you use certain labs. And that is partially because of the extraction method used by the lab. We have strong requirements around phosphorus, and so for Maryland tests, you need to use a lab that's using the MELIC3 extraction because it's a more aggressive extraction method that is going to give you a better sense of how much mobile phosphorus you have in the soil so that you can manage for that. Some of the other labs use modified Morgan, and you know, people can go back and forth and they have different opinions about which extraction method is better or worse, or there's a general thought that MELIC3 is better for southern soils, but up north, the modified Morgan seems to be the more popular choice. So depending on where you are, you might want to look a little more into that. I think we have people from different areas on the webinar, so I'm not going to get too into recommendations on that. Budget is another thing to consider. The test range is from $10 for a very basic soil nutrient test with pH and macronutrients up to like the Cornell test, which is 110 or even more if you go for the advanced test. And then there's various add-ons for a la carte menu for test from each of these labs as well. So it's important to consider what kinds of results you're after. If you really just want to see what you need to feed your crop, a particular crop, for growth throughout the season, or whether you want a broader set of indicators that are going to talk about some of the other soil health principles like the Cornell test. So a few sampling do's and don'ts as you plan for your season. It's best to sample in the fall. And that is partially so that if you have adjustments that need to be made, especially pH adjustments, that you will have time to add the lime and for it to take effect prior to the next growing season. The recommendation is to sample at least every 3 years, but I would say you could certainly do it more than that, but I wouldn't do it less. Definitely plan to use the same lab every time. If you use different labs, then the results will vary a lot. And that just really makes things confusing. It's more important to be able to observe trends over time than to focus on, you have to focus on the specifics somewhat, but trends are also very important to note. And then make sure to make observations about the areas that you're going to test and submit separate samples for the different production areas. So if it's part of a particular rotation where crops may be removing certain nutrients more than others, then you want to sample different production areas separately. Also do a separate sample for problem areas and also areas with different soil types. So the don'ts are mainly the converse of those. One important one is not to sample the soil when it's too wet. And also best to collect the samples at the same time every year, and not to split your samples and send to multiple labs at once, because that will most likely just lead to a lot of confusion. And lastly, don't ignore the recommendations. So you will need some basic materials, a clean bucket. You can use a soil probe, although sometimes you need to collect more samples to composite with a probe because it's just a smaller amount of soil. You can use a regular garden trowel, or you can use a shovel. Consistency is the key when you're actually digging the sample. So if you're taking 10 composite samples using a shovel, you want to make sure that each shovel full is approximately the same amount so that you're not sampling from one area in your field more than another. Other basics: Ziploc bag, Sharpie to write to identify which field you're sampling from, and the soil test submission form. Here's some basic steps. It's good to observe the area that you're going to sample from and kind of make a visual map, make a map to yourself about which pattern you're going to use. It's really best to follow the recommendations. Each lab is going to provide a pretty good set of recommendations for how you collect your samples, but these are some examples of them. For the Cornell test, we use the W or M sample, and we do it in 5 points, but we take 2 samples 10 feet apart at each one of those points. Kind of a random pattern is okay as well. You just want to, again, avoid those outlier areas. You want to avoid any low spots or problem areas in the field. And it will also range. Let's see, I think we have a question popping up.
We do, and that's how soon after you dig the sample do you have to send it to the lab? Can you wait a couple of weeks, or would that affect the results?
Oh, right. Yeah, a couple of weeks is a bit long. Within 48 hours is really the recommendation, depending on how it's being kept. If you're able to refrigerate it or something, you could probably go a little bit longer, but a couple of weeks is a little bit too much. So you're going to want to collect anywhere from 8 to 15 composite samples, depending on how big the area is and what the lab recommendations are, and mix it very thoroughly in the bucket. And then you'll only need to send a couple cups to the lab from each field or sample area. If it is a little on the wet side, you can place it on some paper and let it dry a bit overnight, and then seal it up in your Ziploc bag and label it and send it off to the lab. Okay, so these are some not great pictures, but a little bit more step-by-step of how to actually physically collect your sample. You'll want to remove any plant material off the surface, and you'll see I've kind of made about a 6 to 8 inch wide hole first, and then kind of aim your trowel. If you're using a trowel or a shovel, take a very even, about ½ inch slice going down about 6 to 8 inches, depending. 6 to 8 inches is kind of the norm for the test that we're talking about today. And then throw it in the bucket and just make sure to mix it up as you go along. You'll want to remove any excess soil from the sides of the trowel, again, trying to get the amount that you take from each composite sample to be pretty close to the same. Okay, on to the actual test. This is actually a test from my farm in one of the first years I was in production. It's from A&L Eastern Laboratories, which is now Waypoint Analytical. And it's kind of an interesting test. It's got the basics, so you can see it's got the macronutrients, pH and buffer index, organic matter, a certain number of micronutrients, and then it also has cation exchange capacity and something else. Percent base saturation, which we'll talk about in a little bit. So most basic soil test packages will include pH, organic matter, macronutrients, and cation exchange capacity. That's really the most basic package. So this soil test, what it doesn't have, something very important to crop health is nitrogen. And we'll talk a little bit more about that in a little bit more detail, because nitrogen is the nutrient used in the largest quantity by crops, and yet is one of the most difficult to measure. Another interesting thing right off the bat to note about this test is that my phosphorus was very, very high. And this was right at the beginning of cultivating this land, and that's very typical of soils in Maryland or on the eastern shore of Maryland, where this farm is. So another very important management focus there, phosphorus. And let's see, I was also dealing with, so the beds we're looking at were in a fairly fertile area of the farm. So it's actually got organic matter that's a little bit higher than is typical of sandy soils on the eastern shore, except for in this one area that I have called sand pit, which was literally pure white sand with like not supporting almost any growth. That was something that we wanted to work on. We wanted to see if we could restore this land, to this one huge field, to some kind of productivity. And I would imagine that some of you on this call have had to deal with that somewhere.
We have another question. If a person is unfamiliar with what is a good or bad level of those nutrients for crops, where would you recommend they start looking for those numbers?
Well, it's partially in the, so most of the tests will have some levels, some optimum levels. And a lot of the sites have that. I'm trying to remember if I have a slide further on that shows optimal amounts.
I was gonna say, I know one of the slides, at least one of the tests has a chart that shows whether for vegetable crop production, the nutrients that the test has detected are in low, medium, optimum, or very high levels. And then this test has, if you look, for example, if you look in the left-hand side at organic matter, you can see it says medium, VH for very high, medium, L for low. So most of the time the test will give you parameters. Lisa, is it correct that the different tests sometimes have different ranges of what they consider to be optimum as well?
Well, there's different ranges based on your cation exchange capacity. So if you have a low cation exchange capacity, you will need, the optimum range of nutrients will be higher than if you have a high cation exchange capacity. So that's why, you know, optimum is a little bit of a wiggly number. And the optimum number will also change depending on your crop. So different crops have different nutrient needs. So the range is usually fairly high in terms of what's considered optimum. And when you are sending in a soil test, it's good to identify what crops you'll be growing. For a lot of us growing mixed vegetables, you often say mixed vegetables, and they can give you a range of what the recommendations are, and they'll base it on that. Okay, so I think we can move on to the next slide. Talk about, there we go.
Good question for you, Lisa. Have you ever used like I'm forgetting what it's called. It's like the Mid-Atlantic Crop Manual that has different nutrient recommendations for different crops. And I know Maryland nutrient management plans have those as well. Is that something that you would recommend folks check out if they're trying to learn the nutrient needs of different crops, or are there other resources that you would recommend?
I have not used that in particular, but it sounds like a great resource. So yes, I would say it's great to look at your soil tests, and for something, if you are doing mixed vegetables and you have to kind of balance your soil for the general good but then look at the individual crop needs so that you can do additional side dressing as needed. I mean, different crops are going to need different nutrients in different quantities, no matter what, because you know, if you apply too much nitrogen to your tomato plants at the wrong time, you're going to get a lot of leaf growth and not a lot of fruit production, and vice versa. So you're always going to have to do a little bit of tweaking for each crop, you know, to tailor to their individual needs. So with the soil test, and in a situation like a general situation like that, you just want to go for an overall balance.
Thanks.
And so there are 17 essential plant nutrients, and essential means that they are necessary for the plant to complete its life cycle from vegetative to reproductive stage, and no elements can be substituted. So some of those essential non-mineral nutrients, or essential nutrients are non-mineral, and they come from the air, so carbon, hydrogen, and oxygen. But the ones that are generally included in our soil tests can be broken down into primary and secondary. The primary macronutrients are NPK. And so a lot of your fertilizers and stuff that you see will just have 3 numbers that indicate those numbers of nitrogen, phosphorus, and potassium. And the reason those are primary macronutrients is because those are the 3 nutrients needed in the greatest quantity by for healthy crops. And then the secondary are also macronutrients, because they're still needed in relatively decent amounts. And those are calcium, magnesium, and silicone. So as we said before, nitrogen is not on all of the tests. Sometimes there will be a total nitrogen measurement, but total nitrogen refers to the organic nitrogen that's in the soil, not necessarily the plant available nitrogen. For it to be plant available, it has to be in the form, oh, I'm sorry, next slide. There we go. It has to be in the form of NH4 ammonium or NO3 nitrate. It's most available as nitrate, but it can be absorbed as ammonium. However, then the plant will have to use additional energy kind of internally to turn it into nitrate, which will use up some extra energy. Nitrifying bacteria in the soil convert ammonium to nitrate. One of the reasons it's so difficult to measure also is because nitrate is very mobile, and it is it's very easy for it to leach through the soil and even down to the groundwater. It's also susceptible to denitrification, so it's volatile, and it will end up back in the atmosphere. And it can also become immobile if the carbon to nitrogen ratio in the soil is off, if it's too high. So if you've incorporated a lot of high carbon crop residue into the soil, it's going to take some time for the bacteria in the soil to consume that material, which is going to tie up the nitrogen during that time, and then eventually it will become available again as the bacteria die off, and actually their decomposing bodies will become plant available nitrogen.
Lisa, quick question. We have someone wondering if a soil test would show, I guess, would screen for bacteria and other microbes in the soil. If soil tests typically measure that, and if not, if there's a way to measure that.
There are some soil tests that measure microbial activity in the soil. Not a lot of your standard soil tests do, but tests like the Cornell test or I think the Earth Fort test, bioassays. I think that's another lab that can provide some information about microbial respiration, active protein, other indicators that indicate a lot of microbial activity.
And we have one more question, and that's if the materials listed on the right bind up nitrogen if they're used as mulch on the surface of the soil, or if this is a measure of how they tie up or release nitrogen when they're tilled in.
It's when they're tilled in. Actually, with using as mulch on the surface is actually a really good way to protect the microbial activity of the soil. So it's adding a layer of protection that is going to increase the biological activity. So only when you incorporate it can it get tied up. Okay, next, there we go. So just a little more evidence, a little visual reference of why the nitrogen cycle, why it's very complex, because nitrogen occurs as solids and liquids and gases, and it changes forms a lot, and it's coming and going all the time. So a very important indicator, and even though it's not always included in your lab report in terms of measuring how much you have in your soil, there will still usually be recommendations about how much you should apply. So one thing to keep in mind with that is with nitrogen applications is because it's very volatile, they'll give you a general recommendation of how much to apply, but the plant won't be using all of the nitrogen at once. And so it's often better to split the applications of the nitrogen, hopefully with some knowledge of when the plant will be able to take it up most effectively, because otherwise that unused nitrogen will be subject to leaching or volatilization or runoff. And the same is true for phosphorus. Not as subject to leaching, but very subject to runoff through erosion. So along with nitrogen, it's needed in higher quantities. It's, you know, really critical for plant growth, but it needs to be managed fairly carefully in order to protect water quality. Okay, and micronutrients. So micronutrients are often the next step up in lab tests. So you may have to bump up the cost a tiny bit. It's very well worth it though. Micronutrients they're used in very small amounts, but they're still essential. So they're not micro because they're less essential. They just are not needed in as large volumes. They do tend to, they don't tend to be deficient quite as often. So they're not quite as often a limiting factor on plant growth. Because they're needed in such trace amounts, but still very important to pay attention to and to amend when they are low. Some are more prone to leaching than others. Things like boron, they don't stick around in the soil, so it's something you will regularly need to apply, though in very, very trace amounts. Some can also be toxic if it applied in too great amounts. Boron, again, being a good example of that. Yeah. And okay, another thing that comes up a lot, you may hear people talk about the ratio of calcium to magnesium being really something important to look at and to maintain. The optimum ratio is about 6:1, and I've heard people be pretty particular about that, but research actually shows that a little wider of a range is acceptable. I think on the test that I shared, I was at about a 3:1, so somewhere under optimal, but still functional. And I would say 6:1, so it's more of a ratio to aim for. And how you would do that is you would look at what amendments that you're using, and you would make sure not to, for instance, if you have a lot of magnesium, if you're at a 1:1 and your soil is getting acidic and you're going to need to lime, you would want to use ag lime rather than dolomitic lime. Dolomitic lime has magnesium in it, and so you wouldn't want to exacerbate that ratio problem by adding magnesium along with your calcium. So so basically just something to look at when you're considering what amendments that you want to use in order to maintain a good ratio there. Okay, and the ever popular Liebig's law of the minimum. You'll see this in any soil presentation you ever see. It's basically just stating that plant production plant growth is going to be inhibited by whichever nutrient or growth factor that's absent in the greatest amount. So if you're deficient in an essential nutrient, any essential nutrient, you won't have proper yielding growth, even if all of the other nutrients are abundant. And this could be extended to non-chemical nutrients, be extended to water and light and compaction, to any limiting factor. So you can't look at your soil test and say, well, I'm low in potassium, but everything else looks great, so I'm probably okay. Like somehow they're going to make up for the deficit. Unfortunately, it just doesn't work like that. So on to pH. pH has a major influence on the growth and functioning of soils, soil animals, microbiology, and the plants. It also controls the availability of some micronutrients like iron and zinc and copper. When the soil becomes too acidic, it can cause toxicity of various nutrients like aluminum, manganese. It can cause calcium deficiency and iron deficiency that's induced by aluminum, magnesium, and phosphorus deficiency. So most crops, I think that's on the, we'll wait to get there. So you can see from this chart that most of these nutrients are most available in the 6 to 7 range. And that also happens to be the range at which most crops need the soil to be for proper growth. There's 2 pH measurements on most soil tests. There's the regular pH that measures just the acidity that's in the water, and that's what your crop and your soil biology sees. And then there's the buffer pH, and that looks at how many acids you have held in the soil reserve. So this is going to relate to how many, what you have in your cation exchange, on your exchange that can be exchanged, that can come on and off of the soil particle. And this is going to help the lab and help you know how much lime you're going to need. So a higher buffer index, so if your buffer index is higher than your soil pH, it actually means that your soil is going to react very quickly and easily to your lime amendments. And so you're going to need smaller amounts of it than if you have a lower buffer index. Lower buffer index is going to mean that your soil is more resistant to change. And so you're actually going to need more lime to result in a change in pH. So that's a little bit confusing. Maybe it sounds a little bit confusing as I'm saying it, but it becomes more clear once you have a little more understanding of the cation exchange capacity and the buffering capacity. Any questions about that?
Doesn't look like we have any questions yet, but I bet we're going to have some, or I know I'm going to have some of the weird soil tests coming up later. Oh, and we do have a question. Can we explain how buffer capacity relates to cation exchange capacity?
Yes, that's going to be just a couple more slides from now. So hold tight and we'll dive into that. Okay, so this is just showing the range of soil pH that's optimum for the crops. And as you can see, most of the vegetable crops that we're focusing on are really centered around that 6 to 7 range. So that's going to be important for your crop yields. Okay, so yeah, now we're going to look at the cation exchange capacity and the base saturation. And buffering capacity isn't on this test, but we're going to talk about it anyways. So cation exchange capacity, you can know a lot about your soil right off the bat by looking at your cation exchange capacity. Now, for instance, on this test here, I'm in the, what is it, like 10 to 12 range, which a sandy soil would typically be in the like 1 to 10 range. As you can see by that bottom number, the 2.4 that's in my sand pit, that indicates a lot of sand and very low organic matter. Whereas the other fields, even though it's sandy, I know we had added a lot of compost there, and it also happened to be part of an old horse farm where the horses hung out a lot. And so that area was a little more fertile. And so right off the bat, by looking at your cation exchange capacity, you can have some indication of soil type. And yeah, we'll wait to, there we go. Okay, so one way that cation exchange capacity was described to me that resonated kind of early on when I was first looking at my soil tests is a dinner plate analogy. So if you have a sandy soil, you have a very small plate. And if you have like a clay soil with a lot of surface area and a lot of negative charge that can attract the positive elements, then you have a large plate. And no matter how much nutrients, no matter how much dinner you try to put on your plate, the small plate is only going to hold so much and the rest is just going to fall off the edges. If you have a large plate, you can heap the food on and it's just going to hold a lot more nutrients. So that's a good analogy to think about when you're thinking about cation exchange. It's basically your soil texture has dealt you your plate size, and there's nothing you can really do about it except for increase organic matter. And so if you have a sandy soil with a low cation exchange capacity, no matter how many nutrients you're feeding your crops, it's just can only hold so much, and you're going to lose the rest. So basically, you're going to be losing nutrients into the water, and you're also going to be losing money in the form of fertilizers. So better to manage your soil to either time your nutrient applications so that the plant can take them up quickly, since they're not going to stick around in the soil, or work really hard to increase your organic matter, which is the best way to increase your cation exchange capacity. So base saturation is also linked to soil type and climate. So certain elements are more prevalent in humid regions. Humid regions like we have here are going to be higher in calcium and hydrogen and aluminum. Hydrogen and aluminum leading to soils that tend to be more acidic. And then in drier, more arid regions, they're higher in calcium, magnesium, and sodium, which tend to be a little more basic. Base saturation gives you some indication of how the elements like calcium and magnesium will be available for plant use. And the more sites on the soil particle that are occupied by basic elements, the more alkaline the soil will be. So base saturation has an inverse relationship with acidity. So on this chart here, you can see that on a low cation exchange capacity soil, like a sandy soil, there's not a lot of sites there to hold on to cations. And so if some of those sites are occupied by acids like hydrogen and aluminum, you are just going to have a more acidic soil. You don't have a lot of basic cations in your profile to adhere to the soil particles. So in a higher cation exchange capacity soil, like a clay soil, the amount of your exchange complex that's occupied by base cations like calcium and magnesium. And then buffer capacity. So soils with a higher cation exchange capacity, like clays, also have a higher capacity to contain reserve sources of acidity, and so will resist change more than lower cation exchange capacity, because there's just less of everything in the soil, basically. So soils that are higher in clay need more lime to affect change on pH than sandier soils. Sandy soils react quickly and really strongly, and so you actually have to be very careful not to overdo it. The soil test lab should calculate your lime recommended using the buffer pH information that they've calculated. But if you have really sandy soil, it can't hurt to let the lab know so that they make sure to adjust your recommendation and you don't end up over liming your soil. So for instance, the test that I had didn't have buffer capacity as an indicator, and I'm pretty sure that I ended up over liming when I first started out on that land. Okay, these are a couple of good resources. The soil tome, the nature and properties of soils. If you want to get really textbooky, the Cornell Assessment of Soil Health on their site, they have a manual that's full of amazing information. The Soil Fertility Manual from the International Plant Nutrition Institute is a great resource, and then some other books, kind of science for farmers, teaming with microbes, great information about microbial populations, and then Advanced Biological Farming by Gary Zimmer does a really great job at kind of helping you read soil tests as well. So any questions now? Or is everyone ready for a break?
Feel free to chime in the chat box. And we are going to have a space at the end for more questions. We wanted to offer, if anybody has a copy of their own soil test that they would like to have, you know, analyzed or have questions about live on this chat, there's still an opportunity to send it in. You can email it to me directly. My email is in the chat box, and I'm putting it in again. It's caroline@futureharvestcasa.org. We do have 2 questions. One is if there's any applications for this type of testing in hydroponics or soilless mixes.
Yeah, I think that there are. Certainly, yeah, I'm not sure that the soil test labs do hydroponics testing, but I'm certain that some labs do.
Yeah, if anybody knows the answer to that one, I have no idea either. Feel free to chime in the chat box here. And then we had one other question, which is about on-farm tests of soil quality that don't require a lab.
Yes, absolutely. And I have a slide later on with a list of some qualitative and in-field assessments that you can do on your own. And I highly recommend doing that. I think it's a really great way to get to know like a broader context of the components that go into soil health. So being able to visually observe your aggregate stability, you can do a slake test to see kind of how your soil structure is. If it holds together, it'll tell you if you've got a like all the glues and things that are necessary to hold the soil together. It'll show you if your soil is subject to erosion. So the slake test is great. Doing a worm count, just basically counting the amount of worms in a square foot of your garden is fantastic. Great indicator. Things like the jar test, so you understand your texture. Then there's some funny ones. Yeah, I have the whole list at the end, which we'll share, but there's the, bury your soil, your undies, bury your white cotton briefs and see how long it takes them to break down, things like that.
We also, Amanda just put a wonderful link to the Maryland Soil Health Card in the chat box, and that's a good resource as well. And then, let's see, we had one more question. And then I think after that question, why don't we go ahead and plan for our break, and then we'll get into some more specific examples. So Lisa, that question was, this person has had a few tests come back low on sulfur, but has been told that it's probably not worth adding sulfur to the soil. What's your opinion on this?
I think that sulfur is necessary for plant growth, and it's only recently been kind of getting more attention for that purpose. I feel like the older tests don't always include sulfur as an essential nutrient, but I think that yeah, the newer research is pointing to yes. And it can be messy to deal with, but so one way that I recommend adding sulfur, if you don't want to acidify your soil too much, but you need some sulfur, is to use amendments that are in the sulfuric form. So calcium sulfate, like gypsum, will add some sulfur along with calcium, or potassium sulfate is another example of that.
Great, thank you. All right, why don't we go ahead and do a 10-minute break if anybody needs a moment to run to the restroom, get a snack, anything else, and we'll see you all back at 12, let's say 12:15. Does that sound like a plan? All right, and in the meantime, do feel free to contact me directly with any questions. Chat box too.
Were FDA approved. Pea producers in the United States are very lucky. They have an approved drug in each group, and in the case of the macrocyclic lactones, they have a drug in each subgroup. And just to point out some of the things that are noteworthy about this chart, valbazen is very broad spectrum, the most broad spectrum drug on this table. What's unique about it, it is labeled for the removal of tapeworm segments and also adult liver flukes. So it can kill a couple of things that the other dewormers cannot. What is significant about the macrocyclic lactones is that against external parasites, specifically ivermectin is labeled for removal of nasal spots. Generally speaking, within this group, the avermectins are more effective at external parasites, and the milbemycin, cydectin, is more effective against internal parasites. Cydectin also has the distinction of having persistent activity, which means it provides protection after the initial administration. The other group, of course, is prohibit. Prohibit, you see a box that says variable. You can find different information in terms of how effective the dewormer is against immature worms, which don't lay eggs but suck blood. Then the hypobiotic larva, which is a primary survival mechanism of the worm population. I have read where levamisole is effective against the immature worms and hypobiotic larvae. The other thing that's significant on this chart, the dosages are the exact dosages from the
Hey everybody, we're going to get started again in about 1 more minute.
Okay, should I answer a few of these questions that came up during the break before Linda talks about compost tests?
Sounds like a plan.
Okay. One of the questions was, would Epsom salts work to add sulfur in the same way as the potassium sulfate or the gypsum calcium sulfate? And the answer is yes. Epsom salts are high in both magnesium and sulfur, so they can be a really good amendment. You would just want to consider that calcium to magnesium ratio in your soil tests before adding another source of magnesium. And so let's see. Adding organic matter is, yes, it is good to add organic matter to any soil type. In sandy soils, organic matter is going to increase the cation exchange capacity. So it's going to increase the ability of your soil to hold on to nutrients. It's also going to increase your water holding capacity because sandy soils drain very quickly, and so it can be difficult to keep enough water for crop use in the root zone. So organic matter is going to be really, really beneficial there. It's also going to improve your overall soil structure and actually give it some structure, because sandy soil can be very loose. And then it's going to do a lot of the opposite things with clay soil. It's actually going to loosen. It's going to loosen your soil up. It's going to increase the pore space, allowing more air and water, which is really important for your root growth and for respiration, for the soil biology, and it is actually going to increase the cation exchange capacity even of your clay soils, although that's not really a problem. And let's see. Next question is about wet soil sample, if it's allowed to dry for longer than 48 hours. I think it has more to do, I don't think it'll affect pH. I think it has more to do with the nutrient content because there is living biology in the soil, and so things can just change. The chemistry can change over time. So you just don't want to give it too long for that to happen. And I think the 48 hours is a bit more of a recommendation than like a really hard and fast rule. That will also depend what your soil is being tested for. So if you're doing one of the tests that is going to be measuring microbial activity, that would be even more critical. And recommendation on home soil test probes. I'm not sure. I am familiar with the Lamont test, but I don't really remember the details off the top of my head. Anyone else on the call have experience with those that they could share?
If you do, feel free to enter the answer in the chat box here. All right, so it looks like we're all back. Let's go ahead and move on to how to read a compost test. Linda, let's see, let me find you. And
I'm here.
Oh, great. Thanks.
Awesome. Well, hello again, everyone. I think that this will be a nice little diversion from the great information that Lisa has been providing. And I just want to start by saying that I'm personally pretty new to reading compost tests myself. I've been teaching people about how to compost and being a student of how to compost properly for a number of years. But in terms of seeing what the results of a good composting process is and understanding how it gets sort of communicated in this compost analysis, that's still something that I'm pretty new to. But so I'm hoping that today to just provide you with sort of a basic introduction and sort of a basic primer as to how compost test is laid out, what you might be looking for. And I'll, somebody have a question? No, okay.
Anybody unmuted? I'm trying to make sure everybody is muted, but please do keep yourself on mute when someone else is speaking. Thanks so much.
Yeah, okay. I just wanted to mention that my organization, the Institute for Local Self-Reliance, we're planning on hosting a webinar more specifically focused on compost testing and possibly even one on compost applications later in the summer. We'll certainly keep everybody in the loop on that. So today, just want to give you a quick overview of kind of what you might be looking at, and to mention that there is a difference between a compost test and a soil test. So not all laboratories that do soil tests are also going to do compost tests. And probably much like with soil tests, you get what you pay for. So the more extensive compost tests can cost up to like $350. However, the results that you get back and the sort of interpretation of the results can be really helpful if you yourself are not sort of an expert at reading the results. So, but this is more about reading the results. If you are purchasing compost, especially in a state like Maryland, I'm not as familiar with regulations outside of Maryland, but in order to even sell compost, the person producing the compost and selling the compost is going to have to basically register with the state chemist's office, and they have to do testing of their compost. So there's certain things that are already going to be tested. If you are producing compost yourself, then there's some additional tests that you're going to want to consider, and you yourself are going to want to probably send samples to be tested. And then if you're getting compost from a neighbor, perhaps, who isn't officially selling it, there are just certain things that you're going to want to be aware of. But right now, we are looking at sort of the best of the best compost test results. This is the most information you can get, and it's super helpful. It might be a little bit hard to read. Oh, something that I wanted to do was actually share the link to the test that we're looking at. So if you wanted to look at it more closely, I just put it in the chat box. But basically, this is a publicly available compost test analysis from Frederick County, their solid waste department. And they sent this test to the soil control lab in California. While, as I said in the beginning, there is a difference between what gets tested in a soil test versus a compost test. So not all labs will be doing compost tests. And oftentimes sort of the state labs will do very, very basic compost tests. And there are only certain labs that do this sort of level of analysis. Anyway, so now looking at this test, in the upper left-hand corner, you're going to find sort of all your nutrients listed out and the quantities available. And this is where I would ask somebody like Lisa to be really looking at and helping you kind of understand what this means for your application rates, or just somebody, you know, based on your nutrient management requirements and all that, these are things to be aware of. But in general, compost is recommended to be added sort of as an organic matter amendment, not so much as a source of nutrients, just because your nitrogen does tend to be pretty low. And so if you're adding it just to meet that requirement, then you might run into trouble with your phosphorus, which in our region is particularly a concern. So that's just a note on that. But otherwise, things to see in this upper left-hand corner are your pH, your salts, your soluble salts or soluble nutrients. That's kind of communicated. It's communicated as electrical conductivity, your organic matter, and your C to N ratio. And I'll just note that so in terms of what we learn as composters, what we're aiming for good compost, your pH, you're generally looking to get your compost to fall in between a range of 6.5 to 7.5. That's sort of the ideal range. And if you can see here, the pH is 8.54. So that is relatively high. And if you look further down in the analysis, if you click on that link, they tell you that there's, well, you can see that lime's being added to the compost. So that's just something to consider depending on the crop that you're trying to grow and what the pH of your existing soil is. But in terms of electrical conductivity or your soluble salts, here it is 2.8 millimoles per centimeter. I have no idea if I'm saying that unit right, but that's what I looked up. And you're looking for something less than 5. So in terms of that, thank you, Carolyn, for circling that. In terms of the electrical conductivity for this sample, it looks like it's falling in the right range. For organic matter, you're ideally looking for more than 50% in your dry weight. And here it is over 50, which is great. It's 58.6. So in terms of a source of organic matter, you would be achieving that goal with this compost. And then your carbon to nitrogen ratio. This is basically, it helps as an indicator of how mature the compost is, and how applying the compost to your soil, whether it will bind up the nitrogen. If the C to N ratio is too high, if you have too much carbon that still needs to be broken down, it will temporarily bind up any available nitrogen in your soil to kind of finish the decomposition process. So what you're generally looking for is a C to N of 12 to 15 to 1. So 12 to 1 or 15 to 1. And here we have 17 to 1, which is a little bit high, but it's not anything crazy. So just something to note that potentially for some amount of time, if you apply this compost, it could potentially impact your available nitrogen levels.
We have a quick question about which line do you received, and could you explain the difference between the dry weight and as received columns?
Received line. Okay, so different, this is where working with somebody who knows how to read compost tests is going to be helpful, because certain things you want to look at the dry weight, and other things you want to look at the as received. But for the things that we've been talking about, your pH, I mean, there's only as received. Organic matter, you're going to be looking at the dry weight. Your C to N ratio, it doesn't really matter.
And this dry weight, would they dry it out at the lab, and as received would be how it comes in with the moisture that you send it in with?
Exactly. Yeah. Thank you for that clarification. So some of these values need to be considered as a dry weight, but some things are as they are in the actual compost sample. So the way that a compost sample is taken, there are, specifications as to how you go about doing that. You definitely don't want your compost to sit too long before you send it into the lab, specifically because if you want to know the moisture content of your compost and so on, it can't sit for too long. So that's sort of an overview of the upper left-hand corner of what I'm going to touch on right now. And the bottom left are really going to be your heavy metals. And if you're buying a compost, most likely they wouldn't be able to sell the compost if this heavy metals section had any red flags. So generally this is just, they need to prove that they're meeting the EPA sort of set limits for these heavy metals. And generally speaking, heavy metals are going to be the biggest concern for compost that's being made with biosolids, just because they're dealing with generally anything that goes down the drain. But in general, of course, you want to make sure that whatever compost you're getting ready to use is passing in all of these kind of variables. Then the upper right-hand corner of the test, not every compost test is going to look at stability and maturity, but these are great factors, these are great pieces of information to have. And basically what you are looking at with the stability and maturity is whether the materials that have been put into making the compost are actually done decomposing. So in the left-hand corner of the compost test, you'll actually see ammonia, and you'll also see nitrate. So what Lisa was saying earlier is that the nitrate that you need for your plants, the nitrogen that you need for your plants is going to be in sort of the nitrate form, whereas ammonia will tell you if there's a high level of ammonia available or being measured in your compost, it's still actively decomposing, so it's not mature. And so by sending your compost to a laboratory, they will basically measure the carbon dioxide that's being offset and that's being given off by the compost sample, and the presence of ammonia, which will tell them how far along in the composting process it is. Here, the compost is stable. And in terms of testing the maturity of the compost or its fitness for use, often a bioassay or a seed germination test will be done. Here they used cucumber, which generally is thought to be sensitive to immature compost. So basically, if you were to take 50 or 100 cucumber seeds and plant it into a sample of the compost, what's your germination rate? And if you have a poor germination rate, it tells you that something in the compost is not conducive to growing plants. And immature compost does have phytotoxins, it has sort of organic acids, it's not totally stabilized, which happens towards the end of the composting process when a compost has been cured properly. So anyway, these are things that a test like this show you, but not all compost tests will give you this information. Another thing that not all compost tests will give you are the pathogen levels. But here, this is showing you fecal coliforms and salmonella. Obviously, nobody wants that in their compost, but those are really going to be considerations with compost from manures made with manures and biosolids and so on. Obviously, we like to see that they pass. And then in physical contaminants, that's really the bigger the facility that you're getting the compost from, the more stuff that they're kind of putting into the mix. There is chance of plastic contamination, glass, metal, that kind of stuff, whatever random stuff people end up putting in their bin. And then in the bottom right hand side, you basically have the particle size, and you as farmers would probably have a better sense of this than I do, like what particle size would be good. But generally speaking, the finer your particle size, the more appropriate it'll be for most of your horticultural uses. If it's going to be, if the particles are bigger, then it's going to be more appropriate for mulch. So that's a very big, high-level overview of this. I'm sure there's going to be lots of questions, but I'll leave it there for the moment, unless we want to answer some questions.
This is great. Thank you so much. So yeah, folks, feel free to chime in with questions in the chat box. I'm going to erase my annotations here because they go onto multiple slides. Cool. All right, so while we're getting questions, Lisa, you want to go ahead and start talking about the soil test? And this is the portion where we're going to get a lot more hands-on and talk about some specific tests and methods and things like that.
Yeah, so this is another soil test that was shared, that Amanda from Plow and Stars Farm shared. And it's in a slightly different format. So I think it's good to see some different formats so you know how to kind of translate the information from one test to another, so that this maybe seems familiar when you're looking at your own test. It looks like, let's see, they measured the pH, the phosphate, potassium, magnesium, and calcium. So all your macronutrients, and nope, no micronutrients on this test. So I'm kind of interested. I don't want to. Does anyone have any questions that come up when you look at this sample? If this was your sample, and you were wondering what to do with it, you know, what pops up as a question?
Feel free to chime in, and if the farmer is on the line and wants to add anything too, feel free to jump in as well.
It's just kind of a good exercise to like look critically at this and say, you know, if this was my soil. Amanda has here that their goals were to maintain the pH. The pH is at 6.5, to balance the phosphorus. So it's very low. It's deficient in phosphorus. To add calcium without adding magnesium or potassium, right? Those are in the exceeds crop needs. The calcium, right, is deficient, right? So we mentioned before then that if you were going to lime in this circumstance, you would want to use ag lime versus dolomitic lime, because you wouldn't want to be adding more magnesium. Now, they are making a recommendation to add 2,000 pounds of calcitic limestone in order to increase the amount of calcium in the soil. The soil pH is already at 6.5. And to me, that would indicate a fairly high buffering capacity, because they're not too worried about adding lime to the soil, even though it's already fairly close to the optimal range. And let's see, increase soil organic matter should always be a goal, and increase soil carbon. So
Sorry to interrupt. I just wanted to mention something on this test as well that I thought was really useful, which is in the little messages section. And that's they provide the actual formula to change pounds per acre to pounds per 1,000 square feet. So if you're growing on a smaller acreage or you're really trying to do things bed by bed, specifically for a certain crop's need, that formula can be incredibly useful. Sorry to jump in there.
No, that is a great point. I'm glad you jumped in with that. And definitely was information I used a lot. Yeah, if you're on a small scale. Something else you'll note in the recommendations is that they say these recommendations they're generalized because she designated mixed vegetable crop. And so without specific crop information, they can only give really general recommendations. And again, you'll want to kind of look at the individual needs of your crops. And that's where being able to divide up the amendments by like bed or smaller field plots will be really, really useful.
And I think when the reason that I didn't ask for specific recommendations here is because I was using this to fill out my Maryland Nutrient Management Plan, which does give specific recommendations per vegetable. So that was why I didn't ask for it on this test. Okay. And as it turns out, we're using this for annual forage for livestock anyway, but I think it's, my goals are still the same, even though we're not doing vegetables on this piece. The goals are still trying to get that phosphorus up into the optimum range, calcium closer to optimum, increase soil organic matter. Our soil organic matter was pretty low when we started, and it hasn't. 5 years of intensive vegetable production have not helped. So I'm looking forward to raising that up this year for sure.
That's great. Not really sure what I can add here because I think the recommendations are pretty accurate in terms of having a goal of increasing organic matter. You could do that by adding compost. You know, if you're on a small enough scale. If you're on a really large scale, like if you're growing grains or row crops, that can be more difficult to just add large volumes of compost. The other way would be growing cover crops, high biomass cover crops, green manure crops, so that you're kind of growing your organic matter in place. And another key component to that would be reducing tillage. So in vegetable production, we tend to rely on tillage a lot, but you actually lose a lot of organic matter, and sometimes it can be an ongoing uphill battle to increase organic matter when you're constantly putting it in place and then tilling it and releasing it again. Mm-hmm. So, and that would go for soil carbon as well.
Should we do the next slide?
Sure.
Oh, that just zooms in, so it's a clear picture.
There you go.
Oh, there we go. Okay.
Oh, and it shows you the test methods that they use. That's really nice.
Great. Yep, MELIC 3. So that would be necessary for Maryland nutrient planning. The one-to-one soil water for pH, that's pretty typical. The buffer pH, right? Yeah, okay. That's the buffer pH is that number under acidity, and that's a very low number, which means that adding that amount of lime is not actually going to have a huge influence on the pH, and that's why even though her pH is already in a pretty good range, she could still add lime as a source of calcium without it having a great effect. So that is, here they're calling it exchangeable cations, but on the other test, that would have been the buffer index. So something to consider there, like the terminology could change a little bit from test to test, but they do have it in the notes here that it's the Malick buffer pH.
And we have another question, which is, can you utilize the soil test post-harvest? So for example, if you planted your crops and harvested them without a soil test, but you're getting ready for the next growing season, I believe the answer is that's the appropriate time to take and utilize a new soil test. Is there anything else you'd like to add?
No, I think, yeah, fall, post-crop, fall is a good time so that you have time to do amendments. You also then, so if you need lime, it'll give it time to take effect, but you also then have the opportunity to use less soluble amendments. So for some things, like there's amendments that are available right away, or less soluble amendments that take more time to become active in the soil. And so taking your test in the fall actually broadens your range of amendments. Sometimes the less soluble amendments also tend to be the less expensive amendments, which is nice. Okay, and here we've got another test, another lab, spectrum analytic. What's out of place? So we've got very low pH. Oh, there we go. And very high iron. And that actually very often goes hand in hand. So acidic soils tend to have more iron bound in them. The iron becomes more fixed, but as you raise the pH, the iron kind of flows more freely. It won't stay in the exchange complex as readily. It's not as bound. So actually fixing pH here would have an effect on the amount of iron showing up in the soil.
So I have a question. Since this is my soil test, this is one that went very wrong. And I'm still not sure why. And that's, I've never seen any iron deficiencies represented in any of my plants. So I've definitely seen the effects of, you know, low nutrients, likely because of the pH and nutrient availability in the soil, but I've never seen any evidence of iron toxicity. And so my guess is that something went wrong with the soil test and there's not actually that much iron in the soil, because at this level I believe I should see major issues in my crops, but I don't know, is that true?
Let me see. I have a note here, I think. Okay, let's see, acidic soils. Yeah, I mean, acidic soils are just higher in iron. Was your soil waterlogged at all?
Not that this test is from a while ago, so I don't believe so. And what's really interesting too is that I've got very low iron levels in my well water. And so the water pH is 5.5. Everything around here is quite acidic, and the pH of 5 is actually very much in line with what other farmers in my region are finding in their soils. I'm in Western Maryland in Washington County. The iron level, however, just seemed abnormally through the roof, given, you know, things like the soil coloration and the plant growth and everything like that.
Yeah, I mean, that is interesting, and interesting that you weren't seeing any effects on crop, because excessive iron like that would also tie up phosphorus and manganese. And so you would likely be seeing effects on crops on multiple fronts there.
Yeah, and there's not good evidence of any sort of phosphorus deficiency in most of the growth, except for during cold seasons. So anyways, that's an example for folks of when you get back a soil test, it doesn't make a lot of sense, right? And so this, for me, was a clear indication that I needed to test again.
Mm-hmm. And did you?
No. I'm a very bad farmer, and I actually haven't. So don't be like me. Do as we say, not as we do in my case. And I actually am planning to test the spring right now because I figure better late than never, and I'll do a test in the spring and then start in the fall with my standard every fall testing. But I need to have a better idea of what's going on in the soil. Yeah, and I do. We've got, do you want to do a time check? We've got about 13 minutes left. And so I've got a couple more slides to get through, and then folks continue to have questions. And then I do have some soil here if we have time.
Okay. This last test I wanted to share is the Cornell CASH test, Comprehensive Assessment of Soil Health. And this just kind of shows a broader range of soil health indicators that goes beyond the nutrient levels in your soil and are really equally as important to crop health, but have kind of, the Cornell test is really the main one that actually looks at these other indicators. Things like aggregate stability and water holding capacity. I mean, if you think about these are the physical and biological characteristics that are going to allow your crops to access those nutrients. So sometimes you can be supplying all the nutrients to the crops through amendments and fertilizers, but if your soil structure isn't good, if there's not enough pore space, you know, because of compaction, or your soil is so sandy that it can't hold water, if you're low in organic matter, if you're low in microbial activity, so it's not offering any pest or disease repression, and it's not, you know, helping break down the organic material in the soil to make those nutrients available. Those are all really critical to crop health as well. And so this is a great test for showing that. On this particular test, so you can see that it's mostly good. It's in the high range, but there's a couple problem areas. They're low in phosphorus. Phosphorus being a macronutrient is necessary for plant growth, so you'd want to amend that, of course. And then aggregate stability. And aggregate stability is a physical characteristic that a lot of vegetable farms struggle with, again, because of the reliance on tillage. So these are all just things that you can keep in mind and work towards. And even beyond this Cornell test, we can go to the next slide, I think. There we go. Is like someone brought up earlier on the call, is you can learn a lot about these indicators by just going out and digging around, looking at the soil, making observations, keep good field management records so that you can keep track of your observations over time from year to year and in different fields. And depending on your level of techiness, there are apps like Land PKS that you can use. There's the web soil surveys are incredibly useful. And then things like the NRCS in-field assessments and the Maryland Soil Health Card are really quite comprehensive as well. You can learn a lot about your soil by using those. And then things like the Slake test, you can test your own aggregate stability. And the jar test, which we looked at, and yeah, I think I mentioned these earlier, and there are more. So I would highly recommend doing a little bit of your own self-assessments out there. And here is a list, a very not comprehensive list of labs that you can use for soil testing. And I don't think anybody shared tests today.
I don't have any that have come in, although I am checking my email one last time, just in case anyone has shared anything. Not seeing anything, but we've got a little bit of time for some more questions if folks have them. And in the meantime, I also have a big bowl of soil if you want to talk through live what some things to look for. Yeah, someone wanted to see the labs again. I'll go back to that real fast. Okay, well, while we're waiting for questions, this is about the only time that this makes any sense to do on a video conference call. I ran out during the break to dig up some soil from a plot, so I thought that we could use a little bit of time just to take a look at this and go through live some things to look for, since it's a little bit hard to be out in the field. And I do see that one person is interested in sending a test. So my email is caroline@futureharvestcasa.org. I'm gonna send that to you and you can send it my way. And we can share it. And we do have one more question, which is, do we have any advice on which soil health metrics are meaningful and which labs are best for testing them?
Well, that's a, so there kind of goes back to the beginning of the webinar, and it definitely goes to what you're looking for. So, you know, as a production farmer, you have to be concerned with crop health and yield and things like that in order to be economical. And so nutrient testing is very important. It's an important metric. But I definitely would not override the importance of the physical and biological. So I think aggregate stability is a great indicator for a lot of things. When you think about what happens during, you know, the huge rainstorms, that's a huge threat to crop health as well, just as much as a nutrient deficiency is. If you have waterlogged soil, if your soil is not draining, if there's not enough pore space for air and water to get to your soils, then, you know, you're going to have a crop failure just as easily as you would through a nutrient deficiency. So that is a really important all-around metric, and one that I think is, you know, very agreed upon. And that's something you can test either on your own or at a lab. I think the Cornell lab is the only lab that does that, but I could be wrong.
So we had one other question about if very high levels of calcium and magnesium are something to worry about.
I don't think that either calcium or magnesium are associated with toxicity. But the ratio is somewhat important. So I would pay more attention to that, and also just try to avoid fertilizers that have high amounts of either of those until it's more in the normal range.
All right, and I do have a soil test that just came in, so bear with me 1 second and I will do my best to get that live on the screen. Hang on here. And I also, in the meantime, I'm going to launch our final poll. So, sorry folks, many technical things happening all at once. All right, so that's the post-event survey. So if everyone can just fill that out quickly before you leave, it would be very appreciated. And let's share. All right, let's see. Can folks see this test?
Great.
Let me see if I can get it to zoom out. There we go. You can see everything. All right. So for the person who submitted this test, do you have any specific questions that you wanted us to address? Things that you were looking for clarification on?
Can I just, this is our soil test, and this was done before we did anything to the soil. We just wanted a starting point, and we're converting field to farm. So since this time, we have cover cropped and done numerous applications of compost and trying to build the organic material. But this was a pretty poor, we were pretty sad when we saw this.
And it's from Logan Labs. Okay, great.
Yeah.
And are you seeing changes?
Well, we're growing. So yeah, for sure we've seen changes. We've got worms now, and this was a year and a half ago. I'm trying to remember how long ago this was. At least a year and a half ago, I think. So yeah, I mean, we're certainly not where we want to be, but I can't wait to do a soil test this fall and see what, how things have changed for us.
I'm her sister, I'm her sister, and also I sent the test. It was April 2018.
Okay.
Thanks.
That's great. And actually, I see someone else asked, do you recommend taking a soil test before purchasing? And I would agree, yes, but I would also say that you really, certain things like building organic matter will take time, but you can make major improvements in your soil to support crop health really very quickly. Some of the amendments, and especially if you consider using some of the less soluble amendments that will kind of build up your soil mineral profile over time, that can be a less expensive way to go. So I think just by correcting your pH and then making sure that you're feeding your crops as needed, but also taking those long-term steps like building organic matter through cover cropping and adding compost, you can really shift things pretty quickly.
Cool. Yep, and as Amanda pointed out, getting a sense of the soil type before purchasing so you know what kind of land you're likely to be working with. So is your soil likely to have a lot of clay or likely to be very acidic, or these, do you want to plant a lime your first year, that sort of thing. So we are at 12:59, and I want to respect folks' time, but please feel free to send in any last questions. And Lisa, you want to walk me through doing a jar test in our last minute so folks can see?
Oh. Do you have a jar?
I have a jar.
Okay. So you can just fill your jar about halfway with soil.
All right. I feel like I'm on Bill Nye. All right.
Sounds good.
There's a clear space.
That works. Wait, is that? It looks a little hot. Is it half or a little more?
There's a lot of airspace at the bottom, so it's a little more than half, but there's some big chunks of air.
Okay, this should be okay. Then you want to fill it the rest of the way with water, but leaving enough airspace at the top that you're going to be able to shake the jar and have everything get mixed up.
Yeah.
So leave like
that.
Yeah, that looks pretty good.
All right.
A little hard.
Got it really.
So we're going to run over time, but you actually need to shake it now for at least 3 minutes.
Yes.
At least 3 minutes to make sure that all of the particles are suspended in the water. And what you will notice is that the sand will settle out very, very quickly. And then the silt after that takes a little longer, a few hours, up to half a day. And then the clay can remain suspended for a really, really, really long time. So it could take up to a couple days. You need to put it down and not touch it for a couple days.
Awesome. Well, I'm going to keep shaking it, but I'm going to assume that folks are probably going to want to leave and go do whatever their next thing is they need to do. And so we can post a photo of what the end result looks like in the file with the slides so you can take a look. But that's how to do your own jar test.
All right. Thanks, everyone. Thanks for joining us today.
All right. Thank you, everybody. If you can just fill out the poll before you leave, I'm going to start the 3-minute clock again. Got a jar of mud here now. All right. Thank you, everybody. This is fun.